Measured, precise. Every calorie you burn, every heartbeat you generate, and every cognitive impulse is regulated by this chemical structure, a single molecule of iodine. The human body cannot synthesize this element. It must be actively sourced from the external environment, ingested and absorbed through the small intestine. Once inside the bloodstream, this trace element dictates an immense range of physiological real estate. It maintains the heart's contractile force, governs central nervous system firing rates, and dictates fast-twitch muscle development. Yet the processing center for all of this system-wide control is highly localized. The entire mechanism runs through a single, small tissue cluster located at the base of the neck, the thyroid gland. Tracing exactly how the body captures and weaponizes this specific element reveals the mechanics behind systemic failures like heart failure, developmental stalling, and myxedema comas.
The regulation of human metabolism begins in the brain, operating under a strict central command structure. The hypothalamus continuously monitors the body's energy demands and communicates directly with the anterior pituitary gland. When metabolic output needs to increase, the hypothalamus synthesizes thyrotropin-releasing hormone, or TRH, and drops it into a dedicated closed-loop portal vein that connects straight to the pituitary. The anterior pituitary reads this TRH signal and translates it into a secondary systemic command. It secretes thyroid-stimulating hormone, TSH, out into the general circulation. To understand clinical thyroid disease, we follow this TSH signal as it exits the brain, strikes the thyroid gland, and alters target cell nuclei. Together, the hypothalamus, the pituitary, and the thyroid form a synchronous feedback loop, a biological thermostat that constantly measures and adjusts chemical output to maintain total organism homeostasis. TRH binds a GQ receptor on the pituitary, triggering a cascade that floods the cell with calcium. This forces TSH release. TSH strikes a GS receptor on a thyroid cell, triggering adenylyl cyclase. This drives up CAMP, powering on protein kinase A. The activation of protein kinase A turns the thyrocyte into a specialized biological factory. It aggressively pulls raw materials from the blood at its basolateral border, pushing finished products out its apical border. By utilizing differing receptor pathways, the central nervous system can amplify a microscopic neurological pulse into a massive, sustained chemical manufacturing process. First, the thyrocyte produces a massive precursor protein, thyroglobulin, inside the rough endoplasmic reticulum, dumping it into the colloid storage vat. Simultaneously, a basolateral symporter aggressively pulls raw iodide from the bloodstream. This iodide diffuses across the cell, exiting via pendrin into the colloid. Now, both raw materials, the empty thyroglobulin protein and the raw iodide, are staged in the colloid space, waiting for final assembly. This active transport system allows the body to capture trace amounts of dietary iodine and concentrate it into the high densities required for endocrine synthesis. Assembly relies on thyroid peroxidase, or TPO. First, TPO oxidizes raw iodide into reactive iodine. Second, through organification, it forces this iodine onto thyroglobulin's tyrosine residues. Finally, TPO couples them. Combining a single and double creates T3. Two doubles yield T4. The factory is skewed toward volume over potency. Roughly 80% of the final product manufactured here is T4, with T3 making up the remaining 20%. TPO is the single mechanical bottleneck of human metabolism. Without its three specific catalytic reactions, the entire endocrine thermostat stops. To distribute the hormone, the thyrocyte reclaims thyroglobulin via endocytosis, merging it with a lysosome. Inside, proteolytic enzymes act as biochemical scissors, snipping the protein backbone. This frees the attached T3 and T4, which are ejected into the surrounding capillary network. Because thyroid hormones are lipophilic, they cannot dissolve in blood. They require specialized transport proteins, primarily thyroxine-binding globulin, to carry them through the circulatory system. Despite being the factory's primary export, T4 enters the bloodstream as an inactive prohormone. Reaching a target tissue, local deiodinase enzymes take over. These enzymes violently shear one specific iodine atom off the T4 molecule, physically converting it into the highly potent, biologically active T3. The thyroid gland provides a stable circulating reservoir of T4, allowing individual organs to activate their own energy supply on demand using local diodonasis. Fast-acting hormones like adrenaline bind to the outside of a cell to trigger temporary reactions. T3 operates completely differently. Active T3 penetrates the nucleus, locking onto DNA receptors to initiate mRNA transcription. The first major output is the mass synthesis of sodium-potassium ATPase pumps. As these massive protein channels embed into cell membranes, they rapidly consume ATP and generate heat, driving thermogenesis. The second output is the upregulation of beta-adrenergic receptors, particularly in the heart. This leaves the cardiac tissue highly sensitive to circulating catecholamines, which drives up resting heart rate and total cardiac output. T3 provides a long-term architectural blueprint, forcing the cell to construct the physical machinery that dictates its baseline oxygen consumption and cardiac contractility. When this transcription engine is forced into overdrive, the physiological consequences are severe. This is the mechanism behind Graves' disease, the most common cause of hyperthyroidism. Graves is caused by an autoimmune hijacking of the HPT axis. The body produces an IgG antibody that mimics the shape of TSH, permanently locking onto the thyrocyte's receptors and stimulating continuous hormone synthesis without feedback control. The resulting overtranscription of ATPase pumps causes severe heat intolerance and rapid weight loss, while excess beta receptors push the patient into tachycardia and severe tremors. These specific antibodies also target tissue outside the thyroid, binding directly to the orbital fibroblasts located behind the eyes. These activated fibroblasts proliferate into fat cells and secrete thick concentrations of hyaluronic acid. This increases tissue volume behind the eye and pushes the globe forward, causing the pronounced exophthalmos visible in this image. The systemic symptoms of Graves' disease represent the predictable consequence of an unchecked cellular factory, flooding the genome with metabolic blueprints. The inverse pathology occurs in Hashimoto thyroiditis, the most common cause of hypothyroiditis in iodine-sufficient regions. Here, the immune system deploys CD8+ T cells to target and destroy the thyrocyte follicular cells, physically dismantling the TPO enzymes and the thyroglobulin assembly lines. The immediate physical manifestation of this aggressive inflammation is the development of a painless, symmetrical goiter, swelling outward from the neck. Inside the target cells, nuclear transcription plummets. Stripped of baseline ATPase and beta receptors, the patient suffers from bradycardia, profound fatigue, and an inability to tolerate cold environments. The lack of thyroid hormone also slows the clearance of complex glycosaminoglycans from the dermis. These sugars build up in the skin and trap fluid, causing a doughy, generalized edema known as myxedema. Without the thyroid's constant genomic signaling, the cellular infrastructure degrades and the body's baseline metabolic machinery begins to fail, causing the entire organism to slowly power down completely. Because we have mapped the layout of the thyrocyte factory floor, we can use specific chemical blockades to sabotage the pathway when the system dysregulates. A drug like perchlorate cuts off the raw materials. It acts as a blockade at the very beginning of the pathway, inhibiting the basolateral symporter and stopping iodide from ever entering the cell. Thioniamides strike deeper into the factory. They directly inhibit the TPO enzyme, freezing its catalytic gears and halting both the oxidation and coupling of iodine. Medications like propylthiouracil offer a peripheral line of defense. By inhibiting tissue deiodinases, they prevent the circulating prohormone T4 from converting into the highly active T3 at the target organ. The thyroid is the master transcription engine for human survival,